So here we are. In the background, actually, you can see a photo of the Gulf nearby Trieste, where there's lots of nice hiking routes. So hopefully one day, not too much in the far distant future, you'll be able to visit us, and so we can show you the beauties of Trieste beyond the science. In our lab, we work on two different areas. One is the focuses on peptide-based materials and gels. And I think Jonathan this morning mentioned a collaboration we have with them on these materials. And then in another lab, we focus on nanocarbons for composites. And we've been very fortunate to enjoy a large network of collaborators that really enable us to study these materials for a large variety of applications. And the common thread is both the nanotechnology, because in both cases we really work on nanotechnological nanostructure materials, but also we really strive to work in water and benign solvents as far as possible. So with an eye on sustainability and also on trying to keep a low impact on the environment. So, for instance, in 2018, we had published on a procedure that was applied on macroscopic carbon nanotube fibers, where we used a portable UV ozone cleaner to generate ozone for the controlled oxidation of carbon nanotubes, so virtually creating zero waste. And we also reviewed this year on an open access article, the green approaches to carbon nanostructure production and functionalization. And you can see also at the bottom some examples of the various applications on which we are involved, thanks to the network of collaborators, as I mentioned. So, yeah, so in particular in our laboratory, what we do is the functionalization of a variety of carbon nanomaterials. And we found that indeed, I mean, this is well known for those working in the field, that the problems of standardization and purity of the original materials is always a key issue of concern sometimes, because you can buy some materials and then you get something that is not quite what you expected. And it is extremely important then, because of course, especially for biological applications, the purity and the reproducibility batch to batch of the starting materials, so in particular, in this case, we're talking about the carbon nanostructures, is crucial for also the biological performance and of course, the toxic potential side effects in terms of biocompatibility. So through functionalization, we really strive to strike the right balance between the properties that you find in pristine carbon nanostructures and functionalized carbon nanostructures. So you lose some and then you win some, and it's not always easy to find the right balance there. And we have reviewed this already a few years back on ACS Nano. And what we became interested in is in studying the effects of different nanomorphologies of carbon nanomaterials on the performance of the composites, because sometimes it can get quite interesting and varying effects depending on the intended application. So, for instance, it has been found that carbon nanotube anisotropy can be particularly beneficial for the regeneration of challenging conductive tissues, such as the nerve and the heart tissues, as they allow for a better connectivity relative to non-anisotropic nanostructured materials with conductive cells, elongated cells, such as neurons, but also conductive cells such as those of the heart. So if you have some control over carbon nanotube, not just functionalization, but also orientation in the material, you can gain beneficial effects in trying to mimic the natural tissue, such as the structure of the, for example, of the heart. So yes, this is one example where really the different morphology of the different nanomaterials is crucial. And more recently, thanks to a collaboration with scientists in Rome, and this was a project, part of a project funded by our government, the Italian government, and now it's ended. But in any case, we looked at phosphoproteomics, so potentially for early cancer detection of biomarkers from biological samples, different biological samples. And this refers in particularly to phosphorylated peptides, because phosphorylation is one of those post-translational modifications of biomolecules that really gets altered in disease. So it's a hot topic of investigation to try to find biomarkers and profiling techniques for early diagnosis. And the problem is that phosphate groups are very labile. And very bright scientists really devoted a lot of efforts in trying to devise new techniques for their enrichment and stabilization so they can be detected. And amongst the various approaches, two of the most popular ones are based on interactions with metal, and especially metal oxides. So we're talking about immobilized metal or metal oxide affinity chromatography. And so with this in mind, we worked and developed nanocomposite materials. In the first example, we used graphitized carbon black, which is a relatively popular scaffold in analytical chemistry, or at least it's gaining more attention. It's widely available and relatively low cost. And so here, the way it works is that if you have magnetized nanocomposite, then it can be very handy for magnetic separation. And therefore, if you have a biological sample, you can load materials on the nanocomposite and separate the waste through magnetic separation. And then, for instance, by changing pH, you can elute the bound biomolecules or biomarkers from the nanocomposite. And the interaction, one of the interactions can be, for instance, as mentioned, between phosphates of phosphopeptides and metal oxides, in particular titania, has been very, has shown a very promising performance in this regard. And then you can do analysis, for instance, through tandem LC mass spec equipment. So in this work, we compared nanocomposites with different nanocarbon morphologies. So we had graphene, carbon nanotubes, nanohorns, and graphitized carbon black. And we magnetized the materials, and then we grew titania, nano-sized titania on top, and applied them in phosphoproteomics, as mentioned. And in the first step, we oxidized all the carbon nanomaterials to the same extent to allow for their improved dispersibility in biological fluids. And then we grew magnetite nanoparticles on top, and we didn't really see a big effect on the different nanocarbon scaffold onto the magnetite morphology or physicochemical properties of the nanoparticles. And then in the consequent step, we grew nano-sized titania, and that's where really we saw different effects, templating effects played by the nanocarbon scaffold. And these were also reflected in the performance of the resulting nanocomposites. So for instance, the graphitized carbon black allowed for the highest number of peptide identifications. But in terms of analyte selectivity, especially peptides as opposed to phosphopeptides, because through the purification process, the preparation of the biological sample, it is possible to separate other fractions containing lipids and DNA. So really one of the key issues is the separation between phosphorylated and non-phosphorylated peptides. So in this regard, the graphene oxide had the best performance. And we looked more in detail at this composite, and here you can see in the TAM image a graphene oxide flake, and then the magnetite nanoparticles, these round nanoparticles that appear darker. And then you can see all these little spots, and through a higher magnification, it is possible to see that those are all titania nanocrystals. And in particular, also through a combination of experimental and in silico studies, thanks to the help also by Manuel Malfranco in Sisseco, the University of Aveiro. And this collaboration was enabled by cost, actually. So it started with an STSM. Manuel really modeled the materials and could provide fine details to see how the nucleation over growth of titania was favored on both sides of the graphene oxide, where he modeled different types of oxidized functional groups, such as carboxylic acids, epoxides, and so on. And we think this is really interesting. So this year we also reviewed the topic in another open access article where we discuss of the templating effects of different nanocarbon morphologies on nano-sized titania, and how this can be important for a variety of reasons, including, as mentioned before, the performance of the final materials. But of course, then it is key to really have high purity, well-characterized carbon nanostructures with good batch-to-batch reproducibility. And so, yes, standardization, as mentioned, is key in the process. So concluding, yes, nanocarbon morphology will definitely have an effect on the composite performance. And this is something that is not always obvious to anticipate a priori. We've seen the example of carbon nanotubes for nerve tissue regeneration briefly, and also of graphene oxide for the growth of titania nanocrystals, so ensuring a high surface area for good interaction with phosphorylated peptides, is applied to phosphoproteomics. And that's all for my talk. And here you see a photo prior to the COVID pandemic of our group in Trieste, where I hope to see you someday in the future. And you can see some of the collaborators. I'm thankful to Professor Prato, who's been my mentor in the nanocarbon area in particular. And the group led by Professor Anna Laura Capriotti in University of Sapienza, who was part of this collaborative project sponsored by the Italian government, and Manuel Malfranco at the University of Aveiro, who helped us with molecular models through the cost collaboration of the previous cost action, which was Multicomp. And at the top, we also have our website and Twitter account in case you want to get in touch or start new collaborations. We're always very happy to engage with other scientists. So thank you for your attention.